everybody. I hope you all are doing good. I am Deepika from LearnoHub, a free learning platform where you can study physics, chemistry, maths, biology absolutely for free only at learnohub.com. So students, we started with the chapter number 2 of class 10 biology, that is structure of chromosome, cell cycle and cell division. So in the previous video we talked about chromosomes, we talked about genes, we talked about why do we need new cells, and we discussed in detail about interphase. Now is the time to discuss about the two types of cell divisions, that is mitosis and meiosis. So without any further delay, let's get started. Now we will talk about the two kinds of cell divisions, the main two types of cell division, that is mitosis and meiosis. So in case of mitosis, it is a kind of a cell division in which the cells that are produced are diploid. Say this is the organism, this is the parent cell, right, and its ploidy is 2n. So the daughter cells that will be produced will also be 2n, right? So the ploidy will be maintained. So basically in case of mitosis, the ploidy of the daughter cells is also 2n. They are diploid. And where are they, I mean, where is this done? So this is, you can see this in somatic cells of our body, right? So in somatic cells, mitosis takes place. Mitosis is the cell division that takes place in case of somatic cells. So they are responsible for growth and development of an organism. An organism grows in size, an organism develops. So this mitosis is responsible for growth and development. Now coming to the second type of cell division, that is meiosis. Now in case of meiosis, the cells, the daughter cells which are produced are half the number of the parent cell. I mean, the chromosome number is half as compared to the parent cell. So say this is the parent cell, right? So the ploidy is 2n. When the daughter cells will be produced, the ploidy will reduce to half, right? So this means this is a diploid parent cell and the daughter cells produced are haploid. Correct? So I hope this is clear. And where do we see this kind of cell division, meiosis? So these are generally seen in gamete formation, right? So in case of gamete formation, this cell division, this type of cell division, that is meiosis, is seen. Correct? Now starting with mitosis. So mitosis is a as compared to the meiosis, it is a very simpler kind of a cell division. So what happens in mitosis? So I will tell you. So there is a one parent, right? So there will be a single parent cell. So it will produce the identical daughter cells, right? So the cells, the daughter cells that are being produced are identical, correct? And also it will maintain the same number of chromosomes as there are in the parent cell. So if say there are two, the ploidy is 2n, so the same will be in case of daughter cells. So this is what we learned. Then if you talk about the types of the phases, not the types, but the two phases of mitosis. So it generally happens in two phases. So first one is karyokinesis and the second one is cytokinesis. So talking about karyokinesis in brief, what is karyokinesis? So it is nuclear division, right? So what happens in this type, in this phase of division of mitosis? So nuclear division takes place. Nucleus of the cell, it starts dividing, right? Then coming on to the next phase of mitosis, that is cytokinesis. So over here cytoplasm division takes place. So there is division of cytoplasm, right? So initially if we have a cell, right? So it will have a nucleus. So initially what happens, the first step for division is the cell will start growing in its nucleus. So the nucleus will start growing in its size also, because the DNA, the chromosomes which are inside the cell, they will replicate, right? So it will grow in size, it will double the number of chromosomes that it already has, and then this nucleus will divide into two. Correct? So this is karyokinesis, the first phase. Then comes the cytokinesis. Now what will happen? This cytoplasm will start dividing, and it will lead to two cells. It will lead to the formation of two cells. Correct? So these are the two daughter cells. Now this is 2n, right? The ploidy of this parent cell is 2n, and the ploidy of these two cells is also 2n, that is the daughter cells. The chromosome number is exactly the same in both of the cells, and they are same as the chromosome number is there in the parent cell, right? So these are the two phases. I hope these are clear. Now we will discuss in detail about mitosis. Now talking about the phases of mitosis, so the first phase is karyokinesis, that is the division of nucleus. So in this phase there are basically the four stages. This phase happens in four stages. So these four phases are prophase, metaphase, anaphase and telophase. Correct? So what happens in these phases we will talk in detail. And if we talk about a plant and an animal cell, so they are kind of a similar changes that happens in all these four phases, but we will discuss each of these phases for both the cells, for animal cell as well as for the plant cell. Correct? So now we know that there are four phases in mitosis, right? So what are those? So they are prophase, metaphase, anaphase and telophase. Correct? So now is the time to discuss these stages in detail. So interphase is the phase that we already discussed in our last video. So it is also called as a resting phase. Right? So why resting phase? Because the cell seems to be inactive during this phase, but actually it is metabolically very active. So what all happens in the interphase? So interphase is further divided into three phases, that is G1, S and G2. So in G1 phase there is the synthesis of these proteins and RNA which are required by the cell, and S phase is the phase when DNA is synthesized. So DNA replication occurs. So DNA content is doubled. Then comes G2 phase. So G2 phase is the phase where the remaining content like proteins and RNA, they are synthesized. So since you remember that structure, that bowl having those Maggi noodles, and I had those green colored Maggi noodles, right? So that is the interphase stage. When we see these chromatin threads like this, they are just like those Maggi noodles which are coiled with each other and difficult to separate out, right? So this is basically the interphase. So this is the animal cell and this is the plant cell. Correct? So now this is the centrosome which has centriole. This area here, this is the nucleus, and over here is the cytoplasm. These are the chromatin threads. This is the nucleus, and inside it this round shaped little structure is called as nucleolus. Correct? So in animal cell, this was the animal cell. Now in the plant cell, so now we know, we are studying it from class 8, that there are two kinds of cells, animal cells and plant cells. So plant cells have one extra wall around them, that is called as the cell wall. So the outer wall is the cell wall, right, which is made up of cellulose. Then inside again, here it will be cytoplasm. Now this is the nucleus, this is the nucleolus, and these are the chromatin threads, like those jumbled, those entangled threads. Correct? So this is the interphase when cell seems to be in the inactive state. So we also call it as a resting phase. Correct? So let us see what happens in the prophase. Now, coming on to the first phase of M phase, that is prophase. So if we talk about prophase, so this is what we call it, the first phase of M phase, right? The mitotic phase, correct? So just recall what we studied in interphase. So there are these chromatin fibers which are coiled with each other, which are entangled, and they are difficult to separate out, right? But as soon as the cell enters into the M phase, so there are the changes in these chromatin fibers. So they become thick and short. Correct? So they something appear like this. They become thick and they become short. Now each chromosome has already been duplicated, so there are a pair of sister chromatids that we see, right? So they look something like this. Now they are attached, these sister chromatids are attached to each other at this point, which is called as centromere. Correct? Simple. Now this we also studied that in case of animal cells, so these are the round ones are the animal cells throughout the slide, and the square ones, or you can say the rectangle ones, are the plant cells, right? So they represent plant cells. Now in case of animal cells, so we studied there are centrosomes. Now this centrosomes, it duplicates and it forms two centrioles, and these are the two centrioles. If you see, they start moving away from the each other. And do you see these ray like structures over here? So these are called as what? Esters, right? So these are called as esters, which help in spindle formation. Correct? Now as you see, this is the early prophase, right? So we divide this prophase into two phases, early and the late. Correct? Just to understand better what is actually happening in prophase. So these centrioles, they start moving towards the opposite poles, as you can see in the late prophase. Now the one centriole is here and another is here, and these are the esters which are helping this cell to make these spindle fibers. Correct? So these are what? These are the spindle fibers. But these esters, they are absent in case of plant cells. They have this nuclear membrane. They have this nucleolus and this nuclear membrane. So nucleolus is what? A small, you can say, nucleus of the nucleus, right? So there is a small, this spherical shaped structure inside the nucleus, and we call it as a nucleolus. So as this prophase continues, this nuclear membrane and this nucleolus, it starts disappearing. So can you see here? So now this is the dotted line. So it represents this nuclear membrane is disappearing, right? So the same is happening over here, and these esters have formed these spindle fibers. Correct? So this is what happens during prophase. Correct? So you have to mention about the centrosome. It duplicates, then the centriole starts moving to the opposite poles, then nuclear membrane and nucleolus starts disappearing. Then what happens? These esters, they form spindle fibers. Correct? So these esters form what? Spindle fibers. Correct? Then there is a centromere, which is actually holding the two sister chromatids. Correct? So these are the things that you need to mention in the prophase. Now let us see about the next phase, that is called as metaphase. Now talking about metaphase. So just recall what we studied in the prophase and try to see the diagram of the metaphase, the cells in the metaphase. So the changes are clearly visible, right? So do you remember when we were studying about the cell in the prophase, the chromosomes were all around, right? So some were lying over here, some were here, some were here. It was not in a specific sequence or in a specific area. Correct? But when we see the metaphase, we observe the chromosomes, they are in the equatorial plane. So what is this equatorial plane? So you remember when we used to study the structure of the earth, we used to say, so this is the equator, the line which crosses the center is the equator, right? So this equator, this imaginary line is the equator basically. So in case of metaphase, all the chromosomes, they get lined at this equator. Correct? So you can see over here. Now there are these spindle fibers and there are these chromosomes. These chromosomes are attached to the spindle fiber with the centromeres. Correct? Simple. So this is the animal cell and this is the plant cell. So chromosomes lie along the equatorial plane of the spindle. Correct? And simple and clear. Let's see our next phase, that is Anaphase. Now coming on to the third phase, that is called as Anaphase. So what happens in the Anaphase? So just recall the metaphase. So all the chromosomes were lying at the equator, right? So all the chromosomes were here. So how these chromosomes were attached to the spindle fibers with their centromere. So this is where the next phase begins, when the centromere in the chromosomes, it starts duplicating, right? So this centromere, it divides, and now what will happen when the centromere will divide? It will split the sister chromatids like this. Correct? Now again, these chromosomes, these split chromosomes, these sister chromatids, they are attached to the centromere, and these centromeres are attached in turn to the spindle fibers. Now these spindle fibers will pull these chromosomes away to the opposite poles, as you can see this. So this is the early Anaphase, where the centromere has split into two parts, and now the spindle fibers have started contracting, and when they started contracting, so at that point of time, these chromosomes, they are moving towards the opposite poles, right? So as we can see here, so same is with the plant cell, right? So same is happening, the contraction of the spindle fibers. So the sister chromatids, they separate and move to the opposite poles due to the contraction of the spindle fibers. So what is the reason for this Anaphase, for these splitting of the sister chromatids? So the it is because first the centromere divides, and secondly these spindle fibers, they start contracting. Now as this phase moves on and we reach to the late Anaphase, so this furrow starts building up, right? So basically what is happening now, the cytoplasm is starting to divide. So now the cytoplasm is dividing like this, right? But what happens in case of animal cells? So in case of animal cell, basically this furrow, the cytoplasm is dividing, and this starts building from outside to inside of the cell. But in case of plant cell, it does not happen. Why? Because there is a extra protection layer around the cell, and that is the cell wall, right? So in case of plant cell, so there occurs the formation of the cell plate, which starts from the center of the cell and it moves to the sides, right? So this is how a plant cell divides, right? So what is it? It is basically the beginning of cytokinesis. Correct? So let's study about telophase and the cytokinesis. So the last phase, that is telophase, and simultaneously we will be talking about cytokinesis. So when we were studying about late Anaphase, so we saw the cytoplasm starts to divide. So there occurs a furrow formation, right? So same happens in the telophase. So this furrow starts deepening now in case of animal cells. And what happens in case of plant cell? So there occurs the cell plate formation, which occurs at the equator. So again, what is the equator? The center of the cell. Correct? So the cell plate is laid in the cytoplasm at the equatorial plane. Correct? So this starts the beginning of cytokinesis. Now, as the cleavage starts it progresses basically. So further the cell gets divided into the two parts. Correct? Simple. So we get the two daughter cells in case of animal cells. Now, all the events that are occurring in the telophase, they are exactly the opposite what we studied in the prophase. So what we studied in the prophase, just recall. So, except this furrow formation. Okay. So we see the spindle fibers, they start disappearing at telophase, right? So do you see the spindle fibers over here? No, right? So the spindle fibers, they start disappearing. Now also the chromosomes, which actually became short and thick in prophase, now they again start thinning and they start becoming like those chromatin threads which we see in the interphase stage. Correct? So the same thing will happen in the telophase, right? So it will happen basically in the telophase. So these chromatin fibers, they start becoming those jumbled or the entangled thread-like structures, and the chromosomes are now visible like this. So we also discussed, so there was this nuclear membrane and nucleolus disappearance in the prophase. Both of them starts reappearing in telophase. So can you see this? So there is a nuclear membrane and there is a nucleolus at the center, right? So this is what, this is nucleolus and this is the nuclear membrane. Correct? Right? So the things that happen in the prophase, they will reverse in the telophase. Correct? Now, in case of, if we talk about the plant cell, so the cell will divide something like this. So these are the two daughter cells of this parent cell, right? So again, there is a formation of nuclear membrane, there is reappearance of the nucleolus, right? So the cell plate grows from the center to the periphery, resulting in the two daughter cells, and chromatids turns into chromatin fibers, right? So there were these thick chromosomes which we saw in the prophase, right? So they again will become this entangled thread-like structures when the cytokinesis is being done. Correct? So I hope this is clear. Very simple and very interesting if we go step by step. Correct? Let us now talk about the difference between mitosis in animal cells and mitosis in plant cells. So, as we know, in case of animal cells, there is a single layered wall, right? There is a single layer covering around animal cells, that is the plasma membrane. But in case of plant cells, there is a plasma membrane and there is a cell wall too. Correct? So, this leads to the difference. This leads to one of the differences in mitosis in plant cell and animal cell. So we will start with mitosis in animal cells. So the first point of difference is esters. So as we were going through the mitosis, all the process, all the phases, prophase, metaphase, anaphase, so we saw there are esters which are formed only in mitosis in animal cells, but in case of plant cells, these esters are absent, they are not formed. Then cytokinesis, that is the division of cytoplasm. So how it happens? So in case of animal cells, it starts something like this. First the nucleus will divide, then from outer sides a furrow is made and cytoplasm starts dividing and it completes this portion and then divides into two. So this is how cytokinesis occurs in case of animal cells. But in case of plant cells, say if this is a plant cell, right? So there will be two layers, two coverings, right? So first there will be karyokinesis. So after that, what will happen? There will be a formation of the cell plate. So the cell plate starts getting created from center towards the outer sides, right? And now finally the cell gets divided into 2, right? So this is how the cytokinesis is different in case of plant cells and animal cells. So there is furrowing of cytoplasm and there is cell plate formation in case of plant cells. Then coming on to mitosis in animal cells. So we see mitosis in animal cells throughout the tissues of the body, throughout the body tissues, except for gametogenesis, right? In gametogenesis there happens meiosis, not mitosis. Then coming on to mitosis in plant cells, so it mainly occurs at the growing tips and the sides, say root tips, shoot tips and the sides. So these places, these are the places, these are the sites in the plants where the growth actually happens, right? So at those places mitosis happens, mitosis occurs. Correct. Then coming on to the next, that is spindles. So while going through all the phases, we saw spindles are formed with the help of esters and centrioles. So esters and centrioles both were absent in case of plant cells, but in case of animal cells, these spindles are formed with the help of esters and centrioles. And in case of plant cells, since esters and centrioles are absent, but they have microtubules and which help in the spindle formation. Correct. So these are the basic differences in plants and animal cells related to mitosis, how the mitosis happens in plants and animal cells and how they are different from each other, how these mitoses are different from each other. So now is the time to recall the stages of mitosis. So do you see this picture? We have numbered it from 1 to 6. So we will be recapitulating the stages of mitosis. So let us start with the 1. So 1. So basically this is the cell, right? So this is the nucleus, and inside the nucleus there are these thread-like chromatin structures which are entangled mass, like the entangled thread-like structures, right? So what we call this phase? We call this phase as interphase. So the phase where there is, like, the cell is metabolically very active. Now coming on to the second one. So this is they have marked this as 2 because if you see this, if you compare this with this, now what is happening over here, it is not as black as like this, right? So the chromatin structures, the chromatin fibers, they are not very entangled, but now what has happened, so there it has started becoming like this. The chromosomes have started becoming like this, right? Short and thick. They are attached. The sister chromatids are attached with the centromere. So which phase is this? This is prophase, right? Then coming on to the third point, which is very easy and very easy to identify also. So what we studied about the third phase, that is the metaphase. I mean, the third point, that is the metaphase, when the chromosomes, they lie in the equatorial plane. So can you see this? All the chromosomes are in the equatorial plane, in the center of the cell, right? So which phase is this? This is metaphase. Then we'll come to the fourth one. So if you see this, now we see half the chromosomes are at the one side and half of them are on the other side. So which phase is this? This is Anaphase, where the spindle fibers have contracted and they have broken down these sister chromatids and taken them to the opposite poles. Now coming on to the fifth one. So what is it? Now do you see this line, something, there is a line in the center. So what is happening? Now the cell has entered into the telophase, where the chromosomes are on the opposite sides. I mean, the sister chromatids which were broken, they are on the opposite sides. Spindle fibers have disappeared and there is the formation of this furrow or the cell plate formation, you can say, right? So so which phase will be this? This will be telophase. Correct. Now coming on to the sixth. Now the cell has divided into 2. The nucleolus has reappeared. The chromatin material has again become like chromatin fibers, which are entangled mass, which appear to be entangled structures. So which phase is this? This is the cytokinesis Cytokinesis, when the cell has divided into two, the daughter cells have been produced, right? So this is the cytokinesis, and now each of these cells will enter which phase? Interphase, and again they will start the same process, G1S, G2, and then they will enter the M phase, prophase, metaphase, anaphase, and telophase, and again they will divide in cytokinesis. Correct? So I hope this is clear to everyone. The next that we are going to discuss is significance of mitosis. So mitosis is very important. Now this mitosis, we are studying in so much of detail about mitosis, how it happens, what are the different phases. So it must be of something like it must have some significant role, right? So in mitosis is very important because it is responsible for growth, increase in body cells. So if a body is increasing in size, this means the number of cells present inside the body, they are dividing. The cells inside the body, they are dividing. That is why a body is increasing in size. And how these cells are dividing? By the cell division called mitosis. Second is repair. Now we know that there are many cells inside our body, and there are some cells which get damaged, and they need to we need to repair these damaged cells. We need to repair these wounded tissues also. So we need to renew the cells, right? So how this renewal helps? The cell division, mitosis, helps in the renewal of the cells. Then coming on to the next point, that is replacements. So when we were talking about different types of cells, why do we need cells, how often the body needs cells, the needs to make new cells, right? So that time we studied there are some cells which live throughout the life. They will be born, they will be formed when a body is growing, right? Like the nervous cells. So once they get damaged, they will never be get replaced. But there are some cells which are lost on everyday basis, like the skin cells, the epidermal cells. So they are lost on everyday basis. So these cells need to be replaced every day, right? So we need these cells to be replaced. Now we know about RBCs, how important these RBCs are to us. They have hemoglobin which carries oxygen, right? So these RBCs have a very short lifespan, and our body needs to renew them, replace them after every few hours, right? So the replacement also helps with the process called mitosis, by the cell division called mitosis. Then coming on to the next, asexual reproduction takes place just by mitosis. There is no meiosis, there is no other type of cell division, only by mitosis. A cell grows in size, grows its content, and when the cell is fully mature, this cell divides and splits, right? And this is how the mitosis helps in asexual reproduction. Then next comes is maintains chromosome number. So the chromosome number is maintained only in mitosis. If we talk about meiosis, the chromosome number is reduced to half. But if say this is the cell, and we know that mostly the parent cell, we take them as a diploid, so that is 2n, we represent it with 2n. So when the daughter cells will be produced after mitosis, the chromosome number will be maintained, which is very important, right? So this is the significance of mitosis. Next topic is mitochondria and chloroplast in cell division. So what happens to mitochondria and chloroplast in cell division? So mitochondria and chloroplast, mitochondria in animal cells and chloroplast in plant cells, they have their own DNA and they have their own ribosomes, due to which they are able to divide on their own. They have certain genes which are due to which they are capable of dividing on their own, right? And how do they divide? They simply split them into two parts, and they divide and they produce two daughter cells, correct? So if we take an example of mitochondria, so see if this is the parent mitochondria, so it will split into 2 by simple fission. Correct? So I hope this is clear. So also there are some cytoplasmic ribosomes, and there are certain genes also which guide these mitochondria and chloroplast for division. Correct? Now, talking about the cells, how old are the body cells? We have so many different cells in our body, we have different tissues in our body. So, let us understand how old are our cells. Correct? So, first talking about island cells, then nerve cells and muscle cells. So, these are the cells which last for lifetime, and once they are dead, they will never ever be replaced. Correct? So we were talking about this in significance of mitosis, right? So there are some cells which are never ever replaced in our body. So they are nerve cells, they are muscle cells, and they are island cells. Correct? Then comes skin cells. So skin cells are replaced every week. Now this is our skin. So there is lot of wear and tear because this is the surface of our body, right? So we need these cells to be replaced after every week, so that our skin is able to protect the internal organs of our body. Then next comes RBC. So RBCs, their lifespan is 120 days, and they are replaced very frequently in the bone marrow. Correct? Then our bone cells. So talking about bone cells, so bone cells are replaced after every 10 years, right? So long gap, but they are replaced after every 10 years. Then comes epithelial cells of gut lining. So they last for almost 5 days, and they are replaced after 5 days. Correct? Then coming on to the some other gut cells, so they are replaced every 15 years. Every 15 years, like say if you are 30 years old, I mean you are not, you are very young, but if a person is 30 years old, so this means that person had some other gut cells when he was born. So when he turned around 15, these gut cells were replaced, and when the person turned 30, then again those gut cells were replaced, right? So every 15 years gut cells are replaced. Okay, then coming on to the liver cells. So liver cells generally live for 300 to 500 days, and after that they are replaced. So you can roughly say around 1 year or little more than 1 year. So these cells are replaced on yearly basis, right? So this is all about how old are our body cells. Isn't it interesting to understand, to look into the detail how often our body replaces our cells, right? So let us look at the next topic. Now the next topic is meiosis. So we talked about two types of cell divisions. One was mitosis and one is meiosis, right? So the mitosis, we studied in so much of detail about the different phases of mitosis, but meiosis, we do not need to study the phases in this class. We'll study in the next class. So we will just talk about what is this meiosis, right? So meiosis, if you try to understand this word, so this is taken from the word meon. So meon means to lessen. It is something that we are reducing, so reduction in the number of chromosomes. That is why we called it as reductional division, right? We call it reductional division because there is a reduction in the number of chromosomes. Now where do we see this type of division? We see this type of division in sex cells or in gametes, right? So we know that when these gametes are formed, they are half the number of chromosomes as the parent cell. Correct? So these cells, where does it take place? It takes place in the testes and ovaries in case of animals, and in case of plants, it takes place in anther and ovule. Correct? So now, what is the significant aspect of meiosis? So the number of chromosomes in sex cells is halved. We know that there are 2n number of chromosomes, right? So we represent it as diploid. I mean diploid we represent with 2n, and if we talk about half the number of chromosomes, it means n, that is haploid. Correct? Now this is meiosis. Now talking about meiosis, we do not need to go into the very much detail of this topic as it is not in your syllabus. We just need the introduction part of this topic. So telling you a little more detail, right? So as we saw in mitosis, there are these 4 phases, right? So there will be prophase, metaphase, anaphase and telophase. So these are the 4 phases. But in case of meiosis, the meiosis is itself divided into 2 parts, right? So there will be meiosis 1 and meiosis 2. Now there will again be the same phases. There will be prophase, metaphase, anaphase, telophase, and if it is in the first say in the first part, so it will be prophase 1, anaphase 1, metaphase 1, anaphase 1, telophase 1, and in meiosis 2 it will be prophase 2, metaphase 2, anaphase 2, telophase 2. Correct? But if we go into little more detail, so as simple we see the prophase 1 of mitosis, same is simpler form in meiosis 2 of prophase 2, right? But in case of prophase 1, meiosis 1, prophase 1, this stage is again divided into 5, that is leptotene, zygotene, pachytene, diplotin and dikinesis, right? So these 5 stages make up prophase 1. Correct? So it is not that simple as we studied the mitosis, but studying all these phases is very much interesting, that we will study in the next class. Now talking about the significance of meiosis. So meiosis is also very significant, as meiosis, in case of meiosis, the chromosome number is reduced to half in gametes. So we have studied about meiosis. So it is a reductional division where the chromosome number is reduced to half. So where does this meiosis take place? It takes place in the gametes, right? So in gametes the chromosome number is reduced to half. So what are the gametes in humans? So they are sperms and egg, right? So the chromosome number will be reduced to half, that is they will be haploid, that is n. Correct? Now the second thing is this provides mixing up of genes. Now what happens when the fertilization will take place? So during fertilization, the maternal and paternal chromosome, so what will happen, this sperm and egg will join, will fertilize. So this process is called as fertilization, and what will be formed? Zygote will be formed, right? So when this zygote is formed, so now see, it is made up, so half the chromosomes are received from father, half from the mother, and now the zygote has the chromosomes of both the parents, you mean the genes of both the parents, right? So there is mixing up of the genes. So maternal and paternal chromosomes are mixed, and that is why the chromosome number is half in case of sperms and egg. Now since it will be n, it will also be n, right? So the ploidy will be haploid, and when zygote will be formed, this will be n plus n, that is 2n, that is this organism will be diploid. Correct? Easy. Second is crossing over. So this is the very important thing that we need to understand in meiosis. So let us understand this. So now we are going to talk about a very important term, that is crossing over. So we just studied what happens. There will be a sperm, there will be egg, both of them will fertilize and they will form a zygote, right? So now this zygote has chromosomes both from parents, from mother as well as from the father. Now these maternal and paternal chromosomes inside will combine with each other, right? So now while separating, these forms something, this X-like structures which we call as chiasma or the chiasma, right? Now these are the sites where these you can say the chromosomes or the genes get exchanged, right? So the chromatids, they are exchanging their parts, Correct. So these will be after these homologous chromosomes, they will separate. So we will see, so this paternal chromosome will have some part of this maternal chromosome, and this paternal chromosome will have the part of the maternal chromosome, right? So there is exchange of genes between the two chromosomes, right? So this is called as crossing over, which happens in prophase 1 of meiosis 1. So I don't want you to get confused with all these things, but while talking about significance of meiosis, this crossing over is very, very important. So this X-shaped structure that is being formed is called as chiasma, and here both the chromosomes, they are exchanging their parts, right? And this is how the young ones, they get the young ones get characters from both the parents. Correct? So I hope this is clear. So let us discuss some questions now. Question number 1. In which of the following options the stages of the mitosis have been given in the correct sequence? The first one is prophase, metaphase, telophase, anaphase. So let us understand what are these stages in the correct sequence. So that is P, M, A, T. So prophase, metaphase, anaphase, and telophase. So let us identify the correct option. So these two are eliminated because prophase should be the first one. Now out of these two, prophase, metaphase, but not telophase, it is anaphase. So B is the correct option. Prophase, metaphase, anaphase, and the last one is telophase. Very simple question, right? Let's move on to our question number 2. Question number 2. Synthesis phase in the cell cycle is called so for the synthesis of more of RNA and proteins, DNA and glucose. So where is this synthesis phase? So this synthesis phase we studied in interphase, right? So where in interphase, like interphase has further 3 stages, that is G1, S and G2. So synthesis phase is this S phase, right? So this is the synthesis phase. So what happens in the synthesis phase? DNA, more and more of DNA is synthesized, correct? Simple question. Let's see our question number 3. So talking about question number 3. So question number 3 is fill in the blanks. First is DNA replicates in a dash of the cycle. So where does this DNA replicate? So do you remember we studied about the cell cycle in 2 phases. So first phase was interphase, right? And the second phase was M phase. So in interphase there were 3 stages, right? So it was G1, S and G2, right? So in which phase was this DNA replicating? So it was replicating in S phase of interphase of cell cycle, correct? So I hope this is clear. Then second is mitosis occurs in our dash cells. Which cells? Somatic cells, right? So they occur, mitosis is a kind of a cell division which occurs in somatic cells where the chromosome number remains the same, and that is why mitosis is also called as equational division. Now third one, that is meiosis, occurs only in which type of cells? Very easy. Reproductive cells. It is a type of a division which is called as reductional division. Why reductional division? Because the number of chromosomes are exactly the half of the number of chromosomes that are in the parent cell. So if there are 46 chromosomes in parent cell, so in daughter cells the chromosome number will be 23, right? So these are what? These are daughter cells, right? Now fourth, modern humans have 46 chromosomes. Their sperm and eggs will have dash chromosomes each. How many? 23 chromosomes each. Why? Because in when these sperm and eggs they are being produced, at that time there is a cell division which is called as meiosis, and meiosis is a reductional division, right, as we discussed in the previous question. So the chromosome number is reduced to half. Correct. Fifth is during the pairing of chromosomes, the dash chromosomes come to lie side by side. Which chromosomes? So they are homologous chromosomes. So we call them as homologous chromosomes. Correct. Now, sixth, the two sister chromatids of a paired chromosomes are attached to each other at during dash the process of crossing over. So they are attached at chiasma. Correct? Simple, but we have revised the chapter, we have revised the cell division, right? Question number 4. Shown below are 4 stages A, B, C, D of a certain kind of a cell division, and these stages are not in sequence, right? So we will write over here A, say this is B, this is C, and this is D, right? So we have to answer the questions that follow. So first question is it a plant cell or an animal cell? So looking at the structure, since there is only one layer around them, so it will be what? It will be a plasma membrane. And what kind of cell it will be? It will be an animal cell, because in case of plant cell, there is a cell wall. There is extra covering, right? When we were studying in parallel about animal cell division, plant cell division, we were drawing one extra layer around the cell in case of plant cells. So first is no cell wall present. So this means this is an animal cell. And second is there are no centrosome in case of plant cell, but here you can see these star-like structures. What are these? These are centrioles, right? So centrioles are present. So this means, since centrioles are present, this means this is an animal cell. Coming on to this B part, is it undergoing mitosis or meiosis? So very simple, we just studied about mitosis only, we did not study about meiosis. And the second thing, when we look at these cell divisions, this is a very simple kind of a cell division that we talked about, right? So this is mitosis. When we talk about meiosis, so there are two phases like meiosis 1, meiosis 2, and in case of meiosis 1 there is a long prophase. Like prophase 1 is very long, which in turn has 5 stages. So what kind of cell division is this? This is mitosis. C part, what should be the correct sequence of them 4 stages among themselves? So we have to find out the correct sequence. So let us write PMAT, right? So this is our sequence. So first is prophase. So prophase is the stage when these chromosomes, they appear to be short and thick, and these centrioles, it splits and it starts moving apart. So where do we see these centrioles moving apart? So that is in the B. So the first prophase, this will be prophase, right? Then coming on to the next one, that is metaphase. So what happens in metaphase? All these chromosomes, they align at the equatorial plane. Correct? So where are they aligning in the equatorial plane? So that is C. Correct? So this will be metaphase. Now comes the anaphase. So what happens in anaphase? These chromosomes, they divide. Correct? Now this is the early anaphase. So these have divided. Where are they divided? In D. So this is anaphase. Correct? So let us write the next D. And now what is left? A. So A is what? Now these chromosomes, these spindle fibers, they have started contracting and they are now pulling these chromosomes apart. Correct? So what is happening? So now the chromosomes which are split into 2, now they are moving towards the opposite poles. And when does this happens? This happens in case of telophase. Correct? Okay. Question number 4, D part. Name the stages that should precede the earliest of these stages. So which should be the stage before all these stages? So what happens before this M phase? So what are these stages? This prophase, metaphase, anaphase, they lie in M phase, right? But before that, what happens? The cell is in interphase, right? Where the cell prepares itself for this division, right? So the answer will be interphase. Then draw the stage named above inside the blank space provided. So we have to draw the stage, right? So it will be something like this, and there will be a nucleus at the center, and the chromatin material will be this jumbled up chromatin material, and there will be a nucleolus, and there will be centrosome. Correct? So this is the diagram that we were looking for, right? Interphase. Question number 5. The diagram below represent a stage during the cell division. Study the same and answer the question that follows. A. Name the parts labeled 1, 2 and 3. So what are these parts? So let us write. So which is this part 1? What is it? It is centrosome. Right? What are these dotted lines? These dotted lines are nothing other than spindle fibers, right? Simple. And what is this 3? So they have marked this as 3. So what is it? It is chromatid, right? So what is it? It is chromatid. Now B part, identify the above stage and give a reason to support the answer. Which stage is this? This is very simple. Which stage? This is late anaphase, right? Why? Because now the chromosomes, the centromere has split, the chromosomes have been pulled apart by these spindle fibers contraction, and these chromosomes are at the opposite poles. And this happens when? This happens when the cell is in which phase? Anaphase. Correct? Let us see what is the question number 5, C part. Mention the type of cells in our body where this type of cell division take place. So which type of cell division are we talking about? We are talking about mitosis. And where does this mitosis take place in our body? So mitosis takes place in the somatic cells. Correct? Okay, D part, name the stage prior to this stage and draw a diagram to represent the same. So we were talking about which phase. So this cell is right now in Anaphase stage. Correct? Now, which stage comes prior to this Anaphase stage? So do you remember what was it? PMAT, right? Prophase, metaphase, Anaphase and telophase, right? So what comes prior to anaphase? So that is metaphase, correct? So what we are going to do? We are going to draw a cell, and in that cell, so let's draw these centrioles here, and now we see there are 4 chromosomes. There were 4 chromosomes which are now split into 2 parts, right? So how will this metaphase stage look like? So it is a stage where all the chromosomes, they are in the center, like on the equatorial plane, correct? So what we are going to do? So we are going to make 4 chromosomes, right? Okay. So we will be drawing them like this. And the spindle fibers, where are these spindle fibers attached to these chromosomes? They are attached at the centromere. Correct? So, this is the stage, right? Which stage is this? This is metaphase. Correct? Okay. I hope all the concepts from the chapter cell cycle, cell division and structure of chromosomes are clear to each one of you. Now we will meet in our next video where we will study more concepts from the next chapter from biology class 10 ICSE. So students, keep solving more and more questions and keep brushing up your concepts. So we will meet in the next video. Until then, you all know, LearnoHub, free helper, best hair.